Welcome to Chapter 10 of Bio 112, The Structure and Function of DNA with M.Hook. So in previous chapters, we looked at how cells divide, how they send their DNA to new cells, and how the DNA inheritance patterns work. Now we're going to look at how once we've got our cells, how we get the DNA to actually code for proteins. Okay. So DNA, we know that it has the ability to store genetic information, pass it on generation to generation. So our nucleic acids, these polymers of DNA and RNA, are made up of subunits called nucleotides. You've got the nucleotides bound together by a sugar-phosphate backbone and a nitrogenous base that codes for proteins. So here's the phosphate sugar backbone. You can see it more zoomed in here with the sugar, 5-carbon sugar. In DNA, it's deoxyribose. In RNA, it's ribose, and the phosphate group, and then the nitrogenous base. And remember, for DNA, it's A, C, T, and G. A binds with T, C binds with G, and they form a double helix structure. Thymine T, cytosine C, adenine A, and guanine G. And then in RNA, anytime the DNA has a T, it codes for an A on RNA, and anytime the DNA has an A, instead of having a T being coded for, it's a U. It's uracil. So RNA has U instead of T. Let's talk a little bit about the history of RNA and DNA. There were multiple labs trying to figure out the structure of DNA. They knew that it passed on genetic material, but they didn't know how. There were a couple different competing labs. These guys are Watson and Crick. They were over in England. Watson's from the US. And there was an American lab led by Linus Pauling. And they were all competing to be the first scientists to find out the structure of DNA. They needed someone who could do really good structural work on molecules. They used X-ray crystallography, which was done by Rosalind Franklin. So she's British too, and she was able to look at really small structures using this X-ray crystallography. This is the picture that she was able to get and that they used. Pauling's group had already determined that it was some sort of helical structure, but they thought it was 3 backbones. And what that picture that Rosalind Franklin found was that it was a double helix. And they rushed to publish, and they won. They got in first. They got Nobel Prizes. Except Rosalind Franklin, she didn't get one. She died of cancer at 37 from doing all this X-ray work without knowing about radiation. So what they found was the model of DNA has a spiral double helix. So it's like if you take a ladder and you spin it, and you can still see that double backbones with connections between in the middle, but now it's twisted. And there are lots of different ways to artistically view DNA. You'll have different attempts in your homework where you can see the sugar phosphate backbone and the nitrogenous bases connecting together. And what those connect with is hydrogen bonds. So remember, we talked about ionic bonds where one atom gives another atom an electron. We talked about covalent where they share. And we talked about hydrogen bonds. Those are the weakest, where partial charges on different molecules were attracted together, and that attraction was a hydrogen bond. And how when there's only a few bonds, it's weak, but when you've got a lot, it gets very strong. You can see A and T make 2 bonds, C and G make 3 bonds. That's why you have to have the right base pairs connected together. Let's look at DNA replication now. So we know that we have to send on genetic instructions to the next generation. Let's look at how that's done, whether that be through cell growth or meiosis. The first step of any cell division is to copy the DNA. During interphase, that synthesis phase, you double the DNA. You make 2 exact copies. And what happens is the backbone of the DNA gets split open. You've got DNA helicase and windase that help open it up. And then you start making matching new strands from the original. So this is called semi-conservative replication because here you're saving one of the old strands on each side. So you're saving, out of the 4 new strands, 2, and you're making 2 new ones in yellow. So it's semi-conservative, you're not starting all over, you're keeping some of the old. So there are specific sites, these origination points, where you start DNA replication. And it goes in both directions. And by the end, you've got 2 new DNA molecules. They're exactly identical as long as there are no mutations. And there's a lot of checks in place, especially with DNA replication, to make sure that we don't have any mutations. So if you remember back to previous chapters, we talked about DNA to RNA to proteins. We're going to go more in depth on it now. So the DNA is the genetic code that gets passed on. We send it out into the cell through RNA, and at the ribosomes, we make proteins. We bind amino acids together. So we go from this nucleic acid language to amino acids and make proteins. So remember, the organism's genotype is the code of sequence of DNA. The phenotype is the proteins that are made and what it looks like. There are 2 stages to reading your DNA to making proteins. We go from DNA to RNA and RNA to proteins. First, it's transcription, then it's translation. And these names really make a lot of sense. So transcription, that means to rewrite the same thing in the same language. So we're going from nitrogenous base language to nitrogenous base. So we're just rewriting it, we're transcribing it. And then when that RNA gets edited, it's ready to go out, it leaves the cell. We're translating it into a new language. We're switching from nucleic acids with nitrogenous bases to amino acids and making proteins. So we're changing that language. The one gene, one hypothesis states that for every gene, it codes for a single protein. They used to think it was one gene, one enzyme, but then they realized there are a lot of proteins that aren't enzymes. So we're going to look at those steps from transcription to translation and looking at the steps of how we go from DNA to the polypeptides to proteins. Okay, first, what's the language of nucleic acids? It's those nitrogenous bases, the nucleotide bases. So that A, C, C, G, that's the code. And from that DNA strand, we make matching RNA based on those coding. And so remember, A binds with T for the RNA, except we don't have T in RNA, we have U. So it's U, U, U. C goes with G, C goes with G, C goes with G. Okay, good. And then based on this set of nitrogenous bases, they code for an amino acid. And we'll look at that. So DNA transcription makes RNA. RNA is translated into a sequence of amino acids, which makes a polypeptide. And remember, a polypeptide is a small protein. The bonds between amino acids are called peptide bonds, so a polypeptide is many peptide bonds. Think about this question. What's the correspondence between nucleotides of an RNA molecule and amino acids of polypeptides? So we've seen there are 20 different amino acids, but there's only 4 different nitrogenous bases. How can we get code for 20 different things if we only have 4 letters? We need to have triplets of the nucleotides. So a set of 3 in a row codes for a single amino acid. Those are called codons. And that means that we have more than enough codons to code for all 20 amino acids. It couldn't just be that, A stands for methionine, because then there would only be 4 amino acids. We need to make it more complicated. So 3 in a row makes a codon. And this genetic code, that codon, is shared by all organisms. So we're all very related. So transcription, we're going from DNA to RNA. We read those nitrogenous bases of the nucleotides. We make an RNA template. So it's important we don't move DNA over and have it become RNA. We open up the DNA, we make matching RNA. Because remember, they're different structures. They've got different sugar, they've got different bases with the U. So this is a nice view showing what's happening. So here, we split open the DNA. We've got what's called RNA polymerase. RNA polymer, we're making big RNA pieces, and ACE means it's an enzyme. So this is the enzyme that helps us. In DNA replication, it was DNA polymerase. Here it's RNA polymerase. You open up the DNA, you start making RNA with RNA polymerase, and it adds the RNA nucleotides in. So we're going to go through the steps of transcription, where the RNA polymerase binds on. We have this initiation, elongation, and termination to make a completed RNA sequence. Okay, so first is initiation. That's when we start. You'll have the RNA polymerase attached to what's called a promoter. It promotes RNA polymerase to come in at the beginning of a gene, and we start RNA synthesis. So then we've got elongation. We keep reading the DNA, adding more and more RNA nucleotides until termination. So once you find this terminating sequence, the RNA polymerase detaches and it's done. So now we process the RNA. What do we have to do to the RNA before it's ready to go to the ribosomes? We have to go through RNA processing. So your DNA actually has different codes like exons and introns. We have to cut out the introns. Exons are excited. We read those become the protein. Introns are cut out, and we glue the exons back together. We add a tail at the end and send it out. Now translation. So we've made the RNA, and now we want to convert it into protein. So first you need mRNA. That's the messenger RNA. That carries the message for the code of proteins. There are actually 3 different types of RNA. What we focused on before here, that's mRNA. That's what we're talking about, the messenger RNA. Other types of RNA you have, the first is tRNA, transfer RNA. So what tRNA does is it comes in, it takes the codon of the RNA and matches up with what's called an anticodon. And then on the end, it's got an amino acid. So it comes into the messenger RNA, it matches up in the right spot and leaves an amino acid. And the last part of RNA is ribosomal RNA. Ribosomes are made up of rRNA and proteins. And that's the actual site where making proteins occurs. So here's a nice view showing the mRNA going through the ribosome and the tRNA coming in when it's its turn and adding on amino acids. That's got 3 stages as well: initiation, elongation, and termination. So initiation, we have the first amino acid bind, and it's always methionine. Inside the ribosome, you'll have the ribosome circle it like a cave kind of, and this will keep moving down, and we'll keep having tRNA coming in, bringing amino acids through. And I've actually got a really nice video that will show, I'll give you the link to as well, showing this process.